My name is Shannon Blocker, and I'm a student in Diagnostic Criteria for Plant Nutrition. In this lesson, I will discuss zinc properties, zinc deficiency and symptoms, and zinc fertilization. Zinc is a natural component of the Earth's crust and an inherent part of our environment. Zinc is present not only in rock and soil, but also in air, water, and the biosphere. Plants, animals, and humans contain zinc. Zinc ore deposits are widely spread throughout the world. Zinc ores are extracted in more than 50 countries. China, Australia, Peru, Europe, and Canada are the biggest zinc mining countries. Zinc is normally associated with lead and other metals, including copper, gold, and silver. Zinc deficiency is a global problem affecting both people and plants. These two statistics highlight the problem in the human population. Zinc deficiency is the fifth most important health risk factor for low-income countries. 450,000 annual deaths in children under the age of 5 are due to zinc deficiencies. Notably, several of the areas with soil zinc deficiency occur in lower income countries and where the primary staple crop is low in zinc. These areas correspond in areas where the human population is zinc deficient and results in the problems mentioned earlier. The symptoms of zinc deficiency can be explained by the function of zinc in plants. Yellowing relates to the lack of chlorophyll. Stunting results from low auxin levels, a growth hormone in zinc-deficient plants. Zinc is a semi-mobile element. It functions as a metal activator for plant enzymes, particularly growth regulators. Deficiency first appears on youngest leaves as interveinal chlorosis, a mottling with corresponding reduction of shoot growth. Plants usually produce small, chlorotic leaves on spindly, shorter stems. Environmental conditions that restrict root growth increase the likelihood of zinc deficiency. Soils associated with zinc deficiency are usually neutral to alkaline in reaction. The more alkaline the soil, the greater the need for zinc. Deficiency is particularly notable on crops growing where calcareous subsoils have been exposed to land leveling or erosion. High soil phosphorus levels have been known to induce zinc deficiency, especially in responsive crops. High levels of phosphorus in plants have been shown to restrict zinc movement within the plant, resulting in accumulation in the roots and deficiency in the tops. Therefore, large applications of phosphorus fertilizer may contribute to zinc deficiency in zinc-responsive crops. In corn, a broad band of bleached tissue occurs on each side of the midrib. These stripes begin at the part of the leaf closest to the stalk and appear first on the upper part of the plant. Symptoms are similar to iron deficiency, but iron deficiency is more pronounced on younger leaves. Plant analysis and soil testing are very effective for detecting potential deficiencies. Zinc deficiency creates shortened internodes on the corn stalk. A normal plant, as shown on the bottom, is in contrast to a zinc-deficient plant, shown at the top of this slide. This young citrus tree shows zinc deficiency induced by citrus greening disease. Greening bacteria have blocked the circulation of sap, so that zinc taken up by the roots cannot reach the rest of the plant. Zinc deficiency is also known as mottle leaf and indicates yellow blotches between the veins of terminal shoot leaves, reduced leaf size, narrow-pointed and chlorotic leaves, and the appearance of small green spots in yellowed areas, but the veins remain green, followed by small-sized and misshapen fruits. Internodes are short, giving the shoot a rosette look, and the fruits become small and hard with reduced yield. Zinc deficiency inhibits both vegetative and reproductive growth. In tomato, the deficiency results in shortened internodes, downward curling of the leaflets, and chlorosis. Under severe deficiency, oozing of the cell contents as brown fluid is seen from the leaves. Deficiency in onions shows up as stunting with marked twisting and bending of yellow striped tops. This photo shows zinc-deficient dark red kidney beans. Yellow to whitish areas are between the darker green veins. It is also similar to manganese deficiency, but dark veins are not as prominent. Leaf size may be distorted and irregular. Horticultural crops vary substantially in their response to zinc fertilization. 3 of the above crops, beans, onions, and sweet corn, are highly responsive to zinc. It is also important to note that there are several vegetable and fruit crops that are non-responsive to zinc. One part per million is the dividing line between zinc-sufficient and zinc-deficient soils based on the Kansas State University soil test interpretations. Broadcast applications can correct zinc deficiency for several years. Banded applications at lower rates can be used to correct zinc deficiency for a crop year, but soil zinc deficiency will likely still be present. I've provided an example of zinc rate formula and the calculation. 11.5 and 11.25 are constants in this formula. If you take 11.5 minus the product of 11.25 and the part per million zinc soil test, the answer is the zinc recommendation. In the case of a highly responsive crop with a zinc soil test of 0.6 part per million, the recommendation is 4.75 pounds per acre. Plant tissue sampling is a very effective way to diagnose zinc deficiency. This slide and the following highlight the zinc concentration sufficiency ranges, sampling growth stage, and the plant part sampled for selected crops, which are those with at least a low responsiveness to zinc fertilization. The usual plant range is 30 to 500 parts per million, with excessive or toxic levels at 500 parts per million plus. 30 parts per million is considered sufficient for all vegetables. Three different types of compounds are used in zinc fertilizers. These compounds vary considerably in zinc content, price, and effectiveness for crops on different types of soils. Zinc sulfonate is the most commonly used zinc fertilizer worldwide and is available in both crystalline monohydrate and heptahydrate forms. Other zinc sulfonate forms are zinc oxysulfonate and basic zinc sulfonate. Synthetic chelates are special types of complex micronutrients generally formed by the combining of a chelating agent such as ethylene diamine tetraacetic acid, EDTA, with a metal ion. The disodium salt of zinc EDTA is the most commonly used chelated source of zinc. With their high stability, zinc chelates are suitable for mixing with concentrated fertilizer solutions for soil, fertigation, and hydroponic applications. They can also be used for foliar sprays, but their relatively low zinc content means that repeated applications may be required for moderate to severe zinc deficiency situations. Recent results indicate that zinc sulfonate is a better and cheaper source of foliar spray of zinc than zinc EDTA in terms of correcting zinc deficiency and improving zinc concentrations in seeds and grains. Natural organic complexes, including those which are manufactured by reacting zinc salts with citrates and with organic byproducts from the paper pulp manufacture, such as lignosulfonates, phenols, and polyflavonoids. They are generally less expensive than synthetic chelates, such as zinc EDTA, but are generally much less effective. In summary, we have discussed zinc properties, such as it's a naturally occurring metal. Zinc deficiency is a human health concern, especially in developing countries. Symptoms of plant zinc deficiency include young leaf, interveinal yellowing, and shorter nodes. Zinc fertilization can include soil and foliar applied formulations, and zinc fertilization also occurs through fertigation and hydroponic methods. This and the following slide list the references used in this presentation. These are the photo credits, which are also listed on the slides containing the photos. This completes my presentation on zinc.